We're going to go ahead and get started while people get their food. So if I could have everyone's attention, please. Okay. It's my pleasure today to introduce Dr. Sandra Sanchez-Roige, who will be speaking at today's GBSBC seminar. Dr. Sanchez-Roige is an associate professor at the Department of Psychiatry here at UCSD and the Department of Medicine, Division of Genetic Medicine at Vanderbilt University Medical Center. Dr. Sanchez-Roige has 15 years of experience in psychiatric genetics. She studied psychology at the University Autonomous of Barcelona, received her doctorate in psychology and neuroscience at the University of Sussex, and completed her postdoctoral training in quantitative and statistical genetics here at UCSD. In 2019, she was appointed faculty status in the Department of Psychiatry at UCSD, where she has stayed ever since. Dr. Sanchez-Roige's research focuses on the causal factors contributing to psychiatric disorders and dimensional phenotypes, particularly substance use disorders and other conditions that are characterized by high impulsivity. Leveraging alliances forged across academic, industry, and community partners, she aims to advance precision psychiatry using genetic, genomic, medical bioinformatic, and cross-species translational analysis. She is also an active member of many professional societies and international consortiums on the forefront of precision psychiatry. Since becoming faculty at UCSD, Dr. Sanchez-Roige has received many grants from the Tobacco-Related Disease Research Program and the National Institute of Health, including the prestigious Avenir Award for Genetics and Epigenetics of Substance Use Disorders. She has over 84 peer-reviewed publications, has won numerous distinguished awards, including the 2018 American College of Neuropsychopharmacology Harry June Award, the 2018 NAR SAD Young Investigator Award, the 2021 NIDA NIAAA Early Career Investigative Award, and the 2023 Research Society on Alcoholism Young Investigator Award. Over the next decade, Dr. Sanchez-Roige seeks to integrate the ever-expanding oceans of human genetics data with data from electronic health records and model organisms, with the ultimate goal of unraveling the biological mechanisms of psychiatric disorders and bringing the most promising findings to the clinic. So with that, I welcome Dr. Sandra Sanchez-Roige.
People at home as well. Thank you so much for joining, everyone. Can I get a sense, how many of you are trainees? About 8 years ago, I was a trainee myself, sitting on one of those chairs. And so you can imagine the excitement that I had to be here in front of you, showing you my work and getting your feedback. So I have posed myself almost the Herculean task of addressing in about 50 minutes how far are we from realizing the promise of precision psychiatry, particularly for addiction, by bringing genetics into the clinic. And so just to say that this talk is for everybody, but especially for all the trainees in this room. How about that? Yeah, that's better, right? Oh, okay. Okay, glad that I asked. Okay, that's much better. So to summarize how complex this challenge is, I would like us to first walk into 3 rooms. The first one is a radiologist's office. We can see highly specialized equipment. The second one is a cardiologist's office. Again, highly specialized equipment. But now let's walk into the third office, the psychiatrist's. We cannot spot any single piece of equipment except for arguably the most popular book in psychiatry ever written, the Diagnostic and Statistical Manual of Mental Disorders. Okay, no problem. Glad you cut that. There you go.
Make our friends go away too, hopefully.
Yeah.
Yeah, perfect. Thanks.
How's that? Great. Okay, take 3. This is the DSM. It contains diagnostic criteria for all mental disorders. And the psychiatrist and the patient would be facing each other using words as the most likely proxy for equipment. And the psychiatrist would try to, and here's a third challenge, there you go, would try to pull threads to reconstruct this hairball or portfolio of lived experiences in a single or a few visits. So it is almost expected that prevention, diagnosis, and treatment mechanisms for psychiatric conditions is extremely challenging. In 2015, Barack Obama introduced the concept of precision psychiatry. where alongside these lifestyle and environmental factors, we would bring biomarkers, DNA, into the clinic. And the reality is that genetics have come into the clinic way faster than anticipated. And we have seen some success stories, such as with the development of new treatments or early diagnosis, but we're also grappling, quite frankly, with horror stories, such as with the pre-screening of embryos or identifying opioid addiction with the tools and information that we currently have, led by some companies. And so the way that I see it, perhaps because I spend too much time with Margot, my 5-year-old daughter, I see a lot of parallelism with a short by Pixar called Piper. So this movie begins with a sandpiper bird searching and enjoying its meal, which could be seen as some of the companies on the right side of the previous slide. But here's Piper, or us, at least myself, the field in its infancy, is in the wave of genetics, entering the clinic and feeling paralyzed, tossed into multiple directions. But here's Piper again, and this time, instead of fighting the wave, it stays underneath it. Takes the time to understand the ecosystem. And then something magical happens. Not only it can find the shells, but the biggest ones that not only eats, but shares with others. And so while this is the happy ending that we're all striving for, I will argue that we are a long way from there. So today I will present the most recent advances in the field of addiction genetics and argue when and if we're ready to translate some of this information into the clinic. I have divided that a fourth problem. All right, I'll carry on. So I have divided the talk into 4 blocks. The first one focuses on alcohol, and I will present a carousel of studies that focus somehow on unconventional study designs. Everything okay? Okay, good. You never know. This one? Oh, the more? Oh, there you go. Okay. Go. You did great. Okay. So the second block of my talk focuses on opioids, and I will show you the value of forging collaborations across academic institutions, but also industry partners. The third one is not on any substance, but rather a behavior, impulsivity, and argue how this will facilitate translational research and bring us closer to the neurobiology of substance use disorder. The fourth and last one is on tobacco and uses electronic health records from hospital populations, which I view as the next frontier in the field of precision psychiatry. Okay, so hopefully no more challenges for me. Let's get right in. Substance use disorders have been the focus of all my career, and although this may seem a severe or extreme manifestation, substance use disorders are amongst the commonest psychiatric disorders. The etiology is complex, as all psychiatric conditions are, because it is embedded in a web of environmental and genetic factors. And as I have alluded to, the challenge that we are facing is that treatments are limited and only partially effective. Over the past 7 years, there has been an explosion of genetic studies that are aimed at finding genetic factors that may be associated with substance use disorders. And the bread and butter of such studies are, of course, genome-wide association studies. As many of you, I'm sure, are familiar with, they're simple but extremely effective in mining or scanning along our genome to identify specific regions that may be associated with a trait, for instance, alcohol dependence. And the proponent of GWAS, that we say that we can use this information to develop new therapeutics, assist in risk prevention, and define nosology that is grounded in biology rather than words. That is the pillars of precision psychiatry. But GWAS have a major shortcoming, which is that in order to have sufficiently large sample sizes, we need obscene amounts of data. And of course, over the years, arrays have become more inexpensive to use, which has allowed us to genotype millions of people. Computational approaches to analyze this vast amount of data have also become more inexpensive to use. But what I want to focus on today is to think deeply about the phenotype, because the biology that we're able to extract is heavily dependent on the trait and the population that we choose to perform our study. So how do we measure substance use disorders. We have to go back to this room, to this book. The psychiatrist would ask us a bunch of questions to determine whether we meet certain criteria. And if we carry 2 or more of the symptoms that I list in this slide, we would be diagnosed with a substance use disorder at various levels of severity. So in this sense, this straight DSM diagnosis could serve as our gold standard. We could ascertain individuals with and without a substance use disorder and perform a GWAS. These have indeed been the major efforts led by the largest psychiatric consortium, the Psychiatric Genomics Consortium, or PGC, Substance Use Disorder Workgroup. And here's a picture of us, or the younger version of ourselves, about 7 years ago. And one of the landmark papers by this group was focused on assembling multiple cohorts to characterize alcohol dependence based on the DSM and perform a GWAS. And after much rigor and effort characterizing that many number of individuals, we were only able to reveal one locus, which is peaking here on chromosome 4, of course, the familial ethanol metabolizing enzyme gene. So it became evident that substance use disorders, like all complex traits, are highly polygenic. Of course, as you know, many thousands of genetic variants, which are here, small dots, confer risk, very tiny risk, and that sadly much larger sample sizes would be needed to boost all of this signal above the significant threshold red line. But easier said than done. It is not hard to imagine that these studies that are case-control study designs, they come at a cost, because deeply characterizing individuals is expensive and slow. We may also introduce heterogeneity. To give you an example, based on the DSM, there's over 2,000 unique combinations to receive a substance use disorder diagnosis. We may also have problems of misclassification, because we have to draw a line of separation between these 2 groups, which is artificial, and that can have an impact on statistical power. To complicate things even further, substance use disorders develop in a chronological fashion, and I will be flashing this diagram at least 5 times today to show how important this is to me. You can start counting. From experimental to regular use, compulsive, cessation, and as it is often the case, relapse. And when we're using a binary case-control study design, which is here at the top, we're obscuring the signal that may be relevant at each of these stages, which could have enormous, I think we will all agree, therapeutic value. So it is possible that different genes may have a different role at different stages. So a complementary approach is to break this wall, which was paper thin to begin with, and instead use a continuous phenotype, such as alcohol consumption or our ability to self-regulate behavior. And because they represent domains of normal function, we can measure them in the general population, even in online settings, reaching the obsession of all geneticists, which is the need to obtain very large sample sizes. So it is, of course, a trade-off or a compromise between obtaining a large sample size at the expense of phenotypic depth. I'm very happy to be here today in front of computational biologists primarily, because you would not believe the source of heated debate that this is among psychiatrists. So I believe I will not have to convince you today of the power of these small blue figurines. So back in 2016, with my endless source of inspiration, Dr. Abraham Palmer, the challenge that we wanted to solve was, where can we get high volumes of data that may be relevant to substance use disorders? And the answer was 23andMe. The beauty of working with a genetic company like 23andMe is that they have already genotyped millions of people. And so collecting phenotypic data could be targeted at large scale for a fraction of a cost and quite rapidly. So we partnered with 23andMe, and when we say we partner, more specifically mean we paid them to deploy an online survey that we compiled with input from psychologists. And this survey, as I will slowly unpack today, contained well-established questionnaires that measure different aspects of substance use involvement. So one of the traits that we measured in this survey are the scores from a 10-item questionnaire audit that measures past year alcohol use. We collected 25,000 responses from 23andMe research participants, which back then we laughed at the idea of going any larger than that, but we were, of course, wrong, very wrong. So we ended up combining this data from that of UK Biobank. Sure, many of you know It is an academic collaboration that has completely revolutionized the way that we do genetics, contains genotype and phenotypic data, including audits in 500,000 participants from the UK. So we combined these two data sets and we performed a GWAS meta-analysis of audits, which identified 10 loci, which was 9 more than the inaugural GWAS of alcohol dependence that we reviewed a few slides ago, including the familial ethanol metabolizing enzyme gene here on chromosome 4. In my view, the beauty of this questionnaire is that it can dissect alcohol use from misuse. So for example, the first 3 items measure aspects of consumption, such as the frequency of drinking, the quantity of drinking, and patterns of binge drinking too much too fast. And the remaining items measure problematic consequences of alcohol use, which subsequently we called audit C and audit B. And so we performed 2 separate GWASs of these 2 audit dimensions, audit C, which is here at the top, and audit B at the bottom. And what was clear was that some loci were overlapping, again, ethanol metabolizing enzyme genes on chromosome 4, but that the overlap was incomplete, emphasizing what it has now become almost a signature phrase, that the genetic architecture of alcohol use and misuse is distinct, and emphasizing the value of deconstructing such 2 core symptoms of alcohol use disorder. A natural next question, and one that you may have, is how genetically correlated were these audit dimensions with that of clinically defined alcohol dependence? And to answer this question, we used what back then I considered a completely revolutionary statistical method to perform genetic correlations. Because while I was well-versed on the concept of phenotypic correlations, where you evaluate pairs of traits that are measured in the same individual, with genetic correlations, you compare pairs of traits, but most frequently are measured in different individuals from different cohorts. And this has many benefits because first, we don't have to measure all the traits that we'd like to measure in the same individual, which would be very expensive. And second, we're also partially removing the effects of the environment because we're not using the same individuals and therefore they do not share the same environment. So we calculated genetic correlations between these audit dimensions and alcohol dependence, and on the surface, we identified positive significant genetic correlations. But underneath, when we compare the patterns of genetic correlations with other complex traits, we found quite puzzling distinct patterns of correlations. So for example, audit C was positively, strongly genetically correlated with alcohol dependence, whereas audit C was only moderately genetically correlated with the very same trait, which is perhaps unsurprising because most people can drink alcohol without misusing or becoming dependent. But certainly surprising, and I'm sure that you will be equally surprised now when you see that audit C was negatively genetically correlated with some forms of psychopathology, some health-related outcomes, and positively genetically correlated with socioeconomic outcomes. Whereas these correlations were not observed for audit B, which are here the blue triangles. On the contrary, audit B was positively genetically correlated with numerous psychiatric disorders, as we had anticipated. These results are not very pretty when you're a postdoc. And they are even more terrifying when you realize that this same set of results replicates in independent cohorts much larger than yours, such as drinks per week in a 23andMe and audit C in the Million Veterans Program. So our first attempt at resolving this puzzle was to panic. But we should have not been so alarmed because genetics cannot be studied without their context. What most of these studies have in common is that they use population-based cohorts, such as 23andMe or UK Biobank, which generally include older individuals with higher socioeconomic backgrounds and somehow low rates of alcohol use. Or they use mega biobanks, such as MVP, which also have very unique characteristics, including the fact that they are mainly US male veterans. So disappointingly, with this information, we concluded that alcohol consumption, at least measured in population-based cohorts, is not a good genetic surrogate of alcohol use disorder. But we were not convinced because audit P showed the expected set of genetic correlations, and it was measured in the same cohort than audit C was. So what was it? Another explanation could be attributed to heterogeneity among the individual items. or due to the fact that we constructed audit C and audit B by summing the scores from all of these questions, which of course is mathematically correct. But it assumes that each item, such as frequency or quantity or binge drinking, is equally informative of this construct of consumption that is of clinical interest. And so we speculated that by not correcting or applying weights to each of these audit items, we would be introducing downstream errors in the genetic analysis that we and others had observed. So to test this idea, we used a genomic structural equation modeling, which essentially allowed us to apply empirically derived weights to each of the audit items. So to do that, we first performed GWASs of each of the individual audit items from 1 to 10, and we used genomic structural equation modeling to find what was common across these sets of phenotypes. And we identified 2 latent factors, one of consumption and the other one problems. And even though we can see that they were strongly genetically correlated, they were best represented by distinct factors. Now let's pay attention to item 1, the frequency of alcohol use, because we noticed that this item did not load as strongly onto the consumption factor, and that unlike the other items, it carried a strong genetic residual variance, which we called frequency residual, and that is the villain of the story, as you will see in a couple of slides. So we performed GWASs of these newly derived phenotypes. And of course, the proof is in the pudding, particularly at noon after eating pizza. And so we repeated the same set of genetic correlations, but now using these newly derived phenotypes. And I did stage that beautifully to tell you that, yes, of course, indeed, by applying empirically derived weights to each of the audit items, we were able to remove the previous paradoxical associations. So for example, now consumption in light blue and problems in darker blue were both strongly genetically correlated with alcohol dependence. We no longer observe negative genetic correlations with psychopathology. In fact, some of these associations now became positive. And we no longer observe the positive genetic correlations with socioeconomic outcomes. On the contrary, it was frequency residual that was a culprit, which are here the darker gray bars. We can see, again, these negative genetic correlations with alcohol dependence, psychopathology, and positive genetic correlations with socioeconomic outcomes, suggesting that the previous paradoxical associations that we and others had observed could potentially be attributed to socially stratified differences in behavior, such as going out to drink with friends to socialize, rather than the variance that is directly related to the clinical interest, a trait of interest that we were interested, such as, for instance, drinking excessively or drinking alone. These findings, in my view, are extremely forward-thinking because confounders always find ways to creep in. And these results show that we could use consumption phenotypes measured in population-based cohorts if appropriate statistical methods are in place towards future meta-analysis of much larger sample sizes, as we demonstrated in this study of problematic alcohol use that combined audit data with clinical data in a cohort of over 1 million individuals and that identified 110 novel loci, which again, it's a dramatic increase from that of the first GWAS that we've reviewed together today. To close this first block, however, I want to contradict myself and show you that sample size is not the only element in the equation that we can fine-tune to advance gene discovery. So in this study, Jean Savage very ambitiously set out to extract all alcohol phenotypes with significant heritability from UK Biobank. And again, she used genomic structural equation modeling to stitch all of these phenotypes together. And we realized that they are not all treated equal, that they load onto 4 different dimensions or factors. Again, we identified a factor of consumption that included audit C and other use phenotypes, problems that included audit P and other clinical measures, and 2 new factors such as atypical preference that included drinking fortified wine and spirits, and a new factor that, because we're not marketing geniuses, we called beer preference because it included beer preference, but also very interesting phenotypes such as drinking without meals or reducing drinking due to medical advice. So we performed then GWASs of these colorful factors And we benchmarked the results against that of Hang Zhao et al., the problematic alcohol use GWAS that we reviewed in the previous slide that was very well powered. Of course, it would have been observed to expect that the level of gene discovery could compete with that of a 1 million subject. But a few new laws have emerged, which are here in green. Now, the tables really did turn when we summarize the genetic liability for each of these factors into polygenic scores, and we use them to predict alcohol use disorder in a high-risk sample, in this case, COGA. Because not only the polygenic scores for problems, particularly in beer, were associated with alcohol use disorder in this high-risk sample, but that in fact, they outperform that of these much larger GWAS of problematic alcohol use, which emphasizes, in my view, the power of using granular phenotypes, which will be critical when and if we're ready to translate any of this information into the clinic. The second part of my talk is on opioids. Every 5 minutes, someone dies of an opioid overdose. And so since the beginning of this talk, this is about 5 people that have died. So it is many of us that are committed to curbing this opioid epidemic. And it will not come as a surprise to you when I say that we're interested in studying this transition from initial use to misuse to disorder. And because prescription opioids are widespread in medical settings, we again believe that we could capture this phenotype in a population such as that of 23andMe. So in 2018, we extended our survey to 125,000 people. And one of the questions that we included was, have you ever taken prescription opioids and not as prescribed? And we learned 4 surprising facts. The first one is that 20% of the participants in our cohort reported having never taken prescription painkillers, not as prescribed. Second, the GWAS was underpowered, but the strongest signal replicated that of a much larger GWAS of opioid addiction from an independent study. Third, genetic correlations with other clinical opioid addiction phenotypes were strong and positive. And we also observed numerous genetic correlations with other comorbid traits, including substance use, pain medication, that are trapped in this constellation of dots that not even those with perfect vision can see. And for those reasons, I also don't expect you to see that among these dots, we identified genetic correlations with risky behavior. So admittedly, at that point, we were concerned. We were concerned that maybe we were just picking up on signal that had little to do with opioids, but more to do with risky behavior, such as that people that may be more inclined towards misusing opioids, they may be more inclined towards risky behavior in general. So in a panic, we used statistical tools, brushing over the details, to condition our signal, which is here the gray dots, on risk, which are now here the red dots. And we repeated the sets of genetic correlations, and here I am only sampling the most prominent ones, including opioid use disorder at the very top. And we can see that this had little impact on the correlations that we identified, suggesting that the signal that we captured was primarily related to opioids and not merely risk-taking. So encouraged by these findings, last year, with my partner in crime, Abe, we launched a prescription opioid genetics study in a cohort that will be the size of UK Biobank across multiple ancestries. This survey that Natasha, that is sitting here, has helped us construct, along with other addiction psychiatrists from the Department of Psychiatry here at UCSD. It measures different aspects, as I have claimed, of this transition from first trying opioids to misusing them, including opioid use disorder diagnosis, as well as other crucial factors that perhaps have more of an environmental nature, such as trauma or stress, in a cohort where everyone will have tried prescription opioids at least once in their lifetime. This survey is already out. We are happy to share that we have compiled responses from almost 250,000 of the sample. And the talented graduate student from the BASB program, Jean Gonzalez, who's very embarrassed that I'm pointing a finger to him, has already started analyzing the first tranche of 100,000 responses. And we have never shared those results publicly, so they are hot out of the oven. So here they are. Surprisingly, somehow alarmingly, a very high number of the individuals in our cohort report having a problem or having been diagnosed or treated for opioid addiction. These numbers are not trivial. We also ask about subjective experiences upon first trying prescription opioids. And I would like to maybe get more subjects for this study. Can you raise your hand if you have ever taken prescription opioids when you went to the dentist? Okay, great, many of you. How many of you liked the way it made you feel? Did you feel euphoric? Did you like them overall? Did you feel energized? Maybe one shy hand I saw. Okay. How many of you absolutely hated the way it made you feel? You vomited. Okay, perhaps a few more hands. Okay, so it turns out that the way in which you answer to these questions, we know that from a body of literature from other substances, particularly for alcohol, it is associated with whether you may or may not misuse and a smaller sample that may go on to develop a disorder. And so we ask questions about positive effects, subjective positive effects upon first use of opioids. And we can see here that the main differences are quite dramatic across the groups of those that report having an opioid use disorder, which are here in orange, compared to those that do not in blue. For instance, people that report having an opioid use disorder tend to like prescription opioids when they first try them. They feel euphoric, they feel more energized. On the contrary, the differences in these negative effects that we've reviewed are more modest, perhaps with the exception of itchy, where intriguingly, individuals that report having an opioid use disorder, they tend to report higher rates of itchiness when they first tried prescription opioids. And my biggest revelation was this one, at least to me. The surprise was that the sensitivity to the analgesic effects of opioids, which is the primary indication for which opioids are prescribed, very little differences across these two groups. So Jean took all of this information and used them as predictors to predict opioid use disorder in this sample. And as we expected, when we used the responses from all of these questions, we achieved the highest performance, which is the red bar based on ROC curves. But the second line comes just from a single item. Whether you like prescription opioids shows a performance that is equally almost as including that many questions. And so to me, these results, of course, I'm biased, but they are very powerful because they suggest that we could be screening individuals for their subjective experiences upon first trying prescription opioids and help us identify potentially a small high-risk subset of individuals that perhaps may need further counseling, monitoring, or even a different treatment. So we're very excited about these results. The GWASs are ongoing, so perhaps Jean could be invited in a few months to share the results on that. All right, the third block of my talk, as I promised, is not on any drug, but it's on a behavior, impulsivity. The textbook boring definition of impulsivity is actions or thoughts that are poorly conceived, prematurely expressed, and what is the key is that they tend to result in undesirable consequences. But this is a very broad definition, and in fact, we know that impulsivity can be captured via different ways, such as our inability to delay rewards over time, the inability to stop a response before it is initiated or after it is initiated. And each of these domains have been extremely well characterized in the field of neuroscience. This trait is attractive because it's associated with many psychiatric disorders, including addiction. And so by studying the fundamental nature of this process, which is present in each one of us at a higher or lower level, we can in turn learn about the etiology of substance use disorders. So in 2016 and 2018, another questionnaire that we included in our survey were several well-established questionnaires that measured impulsivity facets. So we included the impulsive behavior scale, which measures 5 different dimensions of impulsivity. And here's an example. I quite enjoy taking risks. And you can see what the answer to that question for you is and compare it against mine. We also included the Barrett impulsiveness scale, or BIS, which measures 3 different dimensions. And another example is I do things without thinking. So we collected, yeah, rarely. We collected 150,000 responses on these 8 impulsive personality traits, and we performed separate GWASs. We learned, as we expected, that impulsivity facets are heritable, which means that the way in which we answered those questions can be partially, with 10%, attributed to genetic factors. These impulsivity facets are genetically correlated, which are shown here in these colorful red bubbles, but this overlap was incomplete. And this was also reflected in the GWAS. Here we can see that GWAS is independent for each of these traits, which are colored in different colors and are overlaid on top of each other. And we can see that different loci are associated with different impulsivity facets, emphasizing what it had been known in the field of neuroscience for a long time, that each impulsivity facet is governed by different biological mechanisms. And yet, there was one locus that was stubbornly associated with many of these facets that is sitting here on chromosome 3 on this tall tower near the gene cell adhesion molecule 2. This gene is fascinating because it is implicated in synapse plasticity, formation, and the most remarkable fact is that we and others have robustly implicated this gene in other GWAS that seem to be related to deficits of inhibition, including a heart-stopping association, perhaps too good to be true, with a rare deletion in the first intron of this gene and ADHD, as identified by Mike Tarkowski at the Center for Genomic Medicine at Mass General Hospital. But here's the challenge that geneticists were facing. GWAS can only identify regions in the genome that these are not causal. We have relatively little information whether the SNPs affect gene expression, and if they do, what is the direction of effect? So to answer partially this question more mechanistically, I'm fortunate to collaborate with multiple scientists across multiple disciplines where we are manipulating this gene in mice, rats, and even patient cells so that we can identify what are the molecular, cellular, and circuit mechanisms whereby this gene affects behavior. So we must continue investing in translational science. We've landed on the final part of my talk. It argues the use of electronic health records to advance gene discovery for a relatively understudied trait at the genetic level, tobacco use disorder. Because while early stages of vulnerability, such as initiation or cigarette consumption, have seen enviable success, as shown by the GScan consortium, 2,000 loci, really enviable. And later stages of vulnerability, such as tobacco use disorder itself, they have been largely unexplored, which we have established is unsurprising because ascertaining individuals is expensive and slow. So in 2020, our colleagues from the Indigo Consortium took a different approach and studied nicotine dependence by using the scores from a 6-item questionnaire, FTND. And despite these immense undertakings that included identifying what we knew, SNPs associated or links with nicotine receptors, it was clear just by looking at these GWAS that yet again, much larger and more diverse samples would be needed to move the field forward. So over recent years, there has been a shift in the field in which electronic health records can be paired with genomic information to fuel genetic studies of mental health traits, which is the premise of the PsychEmerge consortium led by Jordan Smola and Leah Davies, who came to give a talk, I believe, 2 years ago. And soon after the inauguration of this consortium, we formed the Substance Use Disorder Work Group, and we began with a clear goal in mind. Can we prove, or disprove, that we can use EHR data to identify cases and controls for a trait that it's otherwise hard to collect at a large scale, such as tobacco use disorder diagnosis? Admittedly, we used a very simple phenotype that we can extract from EHR, whether someone had 2 or more diagnostic ICD codes for tobacco use disorder. These codes can be derived by psychiatrists, but the catch is that most likely they are simply derived for billing purposes, so they are imperfect. But we wondered if we could show that this trait captures signal that is relevant to tobacco use disorder, it could serve as a cost-effective strategy for genetic studies of this condition. So within 4 months, the team of talented analysts, some of which are here sitting today, Mariela, can you raise your hand? They collected data on tobacco use disorder, ICD codes across different ancestries, conducted GWASs, and then meta-analyzed this data. And we were positively surprised by the number of loci that surpassed this significant threshold line. All of these loci replicated that of the enviable GWASs of the GSCAN consortium, from aspects of initiation, cigarettes per day, cessation, dependence, which was the clearest evidence of the value captured by tobacco use disorder fee codes. This was also revealed not only by this quantity, but also the quality of the signal, as you will see in the next few slides. First, associations with nicotine receptor genes, which of course need no introduction. Second, drugs of abuse are known to activate the mesolimbic dopamine reward pathway, and we can see here associations in or near several dopamine-related genes. Third, substance use disorders have been speculated to be disorders of learning and memory, and we identified signal invariants in a specific gene that encodes for a specific glutamate receptor, and glutamate is, of course, important for modulating reward-related learning and memory. We then mapped those SNPs onto genes via various methods, and Brittany identified that these genes were overexpressed in pathways related to nicotinic-related pathways and dopamine. They were also enriched in the brain in regions such as the frontal cortex and striatum, which have been studied for decades in the field of addiction. Tobacco use disorder risk genes, which are here squished on the right, also showed interactions with varenicline, as we expected to find, because it is the FDA-approved drug for smoking cessation, as well as other compounds that I don't expect you to see in this spider web that all seem to mirror conditions that are highly prevalent with tobacco use disorder, such as the targets of antipsychotics, dopaminergic agents, and antidepressants. We also identified genetic correlations with expected traits, such as smoking phenotypes, with aspects of consumption, dependence, cessation. But if we look closely, these estimates are far from unity, particularly for cigarettes per day, emphasizing what we had speculated all along, that these phenotypes are possibly governed by a different set of common as well as specific genetic factors. We also identified correlations not only with psychiatric phenotypes, but also somatic phenotypes. And these findings also replicated in another sites from the PsychMatch network in what we usually call phenome-wide association analysis. So for this study, what we do is we calculate polygenic scores for tobacco use disorder, and then we examine what other medical phenotypes are associated with genetic liability for tobacco use disorder. And of course, at the very top, as we expected, we identified the phenotypic manifestation of tobacco use disorder, which serve as a positive control. But if we scan down, we can see again associations with psychiatric and somatic conditions. And I'm not sure if some of you now are questioning these results, but sometimes I do get this question, which is, this is all very good, but how much do those polygenic scores tell us that you could not capture with that enviable GWAS of cigarette traits that we reviewed a few slides ago? So we were concerned about that too. And so what we did was we covaried these results on these other polygenic scores for smoking initiation, cigarettes per day, and FTND, which are here the results in green. And we identified that this had, again, little impact on most of these associations, including tobacco use disorder, which emphasizes the value of studying more severe phenotypes, the power of using electronic health records for this type of research. And lastly, we wanted to see as well how well these polygenic scores for tobacco use disorder were associated with other risky or substance use related phenotypes in a younger population. So we use data from the adolescent brain cognitive development cohort, or ABCD cohort. At the particular wave that we studied, this is a longitudinal study, and we used data from 9 to 11 years of age. So luckily, we couldn't capture tobacco use disorder. But externalizing or risky behaviors, which emerge in childhood and are strong correlates of substance use, they could be measured. And we identified indeed that externalizing or risky behaviors were mostly associated with these polygenic scores for tobacco use disorder, as well as other traits related to cognition, other internalizing, as we call them, phenotypes, such as suicide and depression, and other health-related outcomes, such as BMI. And I would like to close by saying that these findings overall suggest the pervasive overlap, not only between psychiatric conditions, but also with somatic conditions, because after all, our genes, as they say, do not seem to have read the DSM, and suggest new ways to potentially define and treat psychiatric disorders. So, are we there yet? GWAS have delivered on their promise. We have accumulated not only hundreds, but thousands of loci associated with psychiatrics, and as we have reviewed together today, substance use related traits. These discoveries have been propelled by an encyclopedic list of factors, including inventive uses of biobank data, generous funding, as well as the persistent donations of patients and participants that donated their data, and also researchers that have, again, persistently worked to solve all of these funding, some of which we have reviewed are sitting here, among others. It is abundantly clear that with newer data sets, such as that of all of us, that not only has array data, but also sequencing data, the number of loci will continue to grow exponentially. But is increasing sample sizes with the hope to obtain more loci all we should be doing? In other words, how are we going to turn these buckets of statistical signal into biological knowledge? If we can break this wall, yes, we may be able to find better targets that could have a treatment and prevention value. And we're working on developing new methods that do not only work at the single gene level, such as with the CADM2 project, but also that they work at the polygenic and even at the single cell level, so that we can have a 3D map of where and when do those genes have their effects. We must also increase diversity. The vast majority of the studies that I presented today are vastly those of European ancestry based on genotype, which clearly does not represent the global population. And it is many of us that are working on expanding data collection across multiple ancestries. And here I am only listing a few, as well as developing new methods so that we can continue striving making equitable science despite the current climate. It is also clear that with clever uses of passive growing sources, such as in this case of electronic health records, and I've listed Psychematch as an example, but of course the dream would be to have a biobank here at UCSD. It is clear that with this newer, larger data from multiple modalities across diverse patients from different environments and contexts will provide novel insights into the etiology of psychiatric disorders. And so here was Viper, and here we are grappling with the question on whether and if we're ready to translate any of these findings into the clinic. And to do that, I think we'll need to spend time building the so-called learning health systems, this sandbox environment that requires collaborations among scientists across multiple ancestries, clinicians, communities, industries, and these are the partnerships, in my view, that will propel precision psychiatry. That's all I have. So thank you so much for coming. Thank you, everyone, joining from home.